So where we left off was to look at, we saw what constitutes that monomer unit of nucleic acids, the nucleotide. So now, those are the monomers, so now we want to look at how they join. Because DNA, as you know, is this big, long, double helix, what looks like a spiral staircase molecule. And RNA is not as long, but will also be sort of a linear molecule, so we're going to see this. So in order for these nucleotides individually to join, they're going to join through dehydration synthesis. And when they join, we now call these phosphodiester bonds. So we've had a lot of different bond names for each of these processes for all the different macromolecules. And this is the last one. So phospho, you can see the word phosphate in there, right? Diester, which might sound familiar to you too. And the linkage between the individual units of these nucleotides is going to be between the 5th carbon on one of the sugars of the nucleotide and the 3rd carbon on the next one. So they actually link in a very particular order, and you're going to see that, and this is called the 5' to 3' direction. So here it is. So we have, here is one of those units, right? We can see it right here. And here it is linked to another one. So here are these monomer units, these nucleotides. There's one, and there's another one. Now we're going to hook on this third one that's on the bottom. So you can notice a couple things here. Number one is you notice that on this sugar, on this number 3rd carbon or 3' part of this sugar, it has a hydroxyl group, or that thing that you see highlighted, the OH. Whereas the phosphate group on this one that hasn't yet attached, there's the hydrogen. There's our water. That's what's going to be removed by dehydration synthesis, and that's where that bond is going to form. So if you follow it from the left to the right, what you see is this new unit, this new nucleotide, has now joined right here from the 5' end of 1, which is the new unit right here, and it's going to connect onto the 3' of the one above it. So we see this 5' to 3' orientation of the molecule as it's growing this way. Also notice here's the water, so it's right over here, and that this new bond now we're going to call a phosphodiester bond right here. Now you might see the ester, if you remember the esters from the triglycerides. They were an oxygen that was kind of like linked to two other things, so it's like right here. So this is, and that's actually been true of all of our bonds, so they look very familiar, but this is specific to this linkage of a phosphate group now to this carbon of the ribose sugar, this 3rd carbon right above. So this is creating a polymer, right? It's getting bigger and bigger. Here we have, what happens though is in DNA we actually have 2 strands, not just a single strand, but another strand, because remember we said it's like a spiral staircase. So the other side of that is going to run in the opposite direction. That's called anti-parallel. So they're both going to be lined up next to each other. One is going to look like it's going from 5' to 3', and the other one is also constructed in a 5' to 3' direction, but it's inverted. So you're going to see it, it's going to run next to it in this anti-parallel way. So we'll see the 3' end going, and the 5' end, they'll be going in opposite directions. This picture shows that a little bit. I've got some others that I'll show you again. So the other thing that's unique in DNA, which is really the code, it's understanding the code, is understanding what is in what order things are pairing and with whom. So if you look up here at the top, so these are 2 strands. Notice that this is the 5' here, and here's the 3 on this one strand. And then the other strand on the other side is running backwards, so 5' is on the bottom going this way. So what actually pairs? are the two strands. Once you have these two strands, the bases are going to pair. Remember these bases where we have pyrimidines and purines, and they are going to link up, and they're going to link by forming hydrogen bonds. So these are hydrogen bonds. This one is probably very clear because you can see the oxygen and the hydrogen here. This is also a hydrogen bond here between this nitrogen and this hydrogen. So that it's not always just oxygen. Nitrogen is also one that forms hydrogen bonds. But here's the hydrogen, right? So adenine, A, and T always pair up. They're the only two that are going to make this pairing. Where guanine and cytosine, they always pair. So they're always going to be a partner. Guanine and cytosine, thymine and adenine. And the other thing that you might notice is that on this one down here, this guanine and cytosine pairing, we have 3 hydrogen bonds holding it together. And that has some significance, but I'll tell you later. Where this A to T, or T is the double, has 2 hydrogen bonds. So these are complementary base pairs. T and A, or A and T, always bond together, and they're always going to form a bond between them with these 2 hydrogen bonds holding them. Cytosine and guanine are going to do the same, always, C and G, but they're going to have 3 hydrogen bonds. Now, part of the significance of this idea of the hydrogen bonding, remember that hydrogen bonds can be broken, where the covalent bonds are stronger. So the phosphodiester bond that was making the original long strands, that's a covalent bond. These hydrogen bonds they can hold pretty well, so they do, but they also have an ability to break, and we'll see why that's important when we look at the structure of DNA. But at any rate, this is complementary base pairing. Thymine and adenine, cytosine and guanine. So here's another picture. I think, so if you see this on the right, you can see this is the double helix, this spiral staircase of a DNA molecule. And there you see in code with the A's, the C's, the T's, and the G's, the abbreviations for these different bases. Remember, these are nitrogen base pairs, or nitrogenous bases. Just means they have nitrogen. So all of them have deoxyribose sugar, because this is DNA. So every one of these is deoxyribose. We go from, on one side, we're going from a 5', where you see the phosphate sticking up at the top, to the 3' this way, but it's anti-parallel, so on the opposite side, it's 5 to 3. 2 hydrogen bonds between the pyrimidines, and 3 hydrogen bonds between the purines. Okay, sorry. So this, I just kind of made that very unclear. This is the pyrimidine purine. Pyrimidine purine. So I always kind of think of it as the single one is kind of like the pyramids. You know, it's just a single structure. The purine is going to have the double ring. So you get it, this end product is this double helix. And the outside of the helix, so the things which are like the ribbons that you're almost seeing here, those are the sugar and the phosphates of that nucleotide, where the bases are the ones in the middle, and they're the ones that are doing hydrogen bonds. When we use DNA, the DNA remembers the code, but it can't leave the nucleus. So the DNA is in your nucleus. Most cells have it in the nucleus. And in order to get the information, in other words, to make a protein by a particular gene getting expressed, then the DNA has to, or a piece of the DNA, the part that has the gene on it, has to be made into something called RNA. And this is going to be a messenger that's going to be able to take that information and take it out of the cell and then take it to the structures called the ribosomes, which are also RNA, to start putting together all those amino acids in the right order. So in other words, creating that primary structure of the protein. And then from there, all the things you know about proteins folding happens. Now, the thing about it that's interesting is that when RNA is made by copying a section of DNA, it's made, but RNA is going to be just a single strand. It's not going to be a double strand. So just 1/2 of that DNA, I'll go back here for a sec to here, 1/2 of this is going to act as a template for the RNA that's going to be made. Now, when mRNA, which is the messenger RNA, is made, it's basically going to be a copy of that 1/2 of that DNA. So if this is the DNA sequence that has now been opened up so that the copy can be made, and that one side of it has these base pairs, so adenine or A, T, T, G, C, G, C, then the new strand, which is going to be the messenger that's going to get out of the nucleus, is going to complement this. So normally adenine on DNA would pair with T, it would be thymine, but this is where that other one I told you comes in. So this is uracil, which will always replace the thymine. So in mRNA, you're not going to find any T's. Instead, substituting for the T will be U. So where this would normally pair with a T, now we're seeing this A is pairing with a U. This next A is also pairing with a U. Because this T is in DNA, then RNA is going to have A, C, G, C, G. And we're going to do some of this in class. We'll do some practice on this. I think it'll be useful to understanding that. So the difference between RNA and DNA is that RNA has ribose sugar, has these bases, these nitrogen bases, and is a single strand, where DNA has deoxyribose, has all of the same ones except for it has thymine and not uracil, and it is a double-stranded molecule. Oops, sorry about that. So we've mentioned this before. There's something called the central dogma in biology, which basically says the DNA is going to make RNA, and RNA makes proteins. So this is the key to all of the things that we know really in biology, is that it's in your DNA is the instructions that we will make a copy of, create it into some form of RNA, and ultimately translate that into a type of protein. And that was brought into our understanding by Francis Crick. So you may remember Watson and Crick from your past studies, who were the ones that were credited, got the Nobel Prize for figuring out the structure of DNA. RNA is going to create proteins, and I think this is maybe a little bit more than we need here, but just to show you that on a chromosome we have this piece of DNA that's going to get translated or transcribed, not translated, transcribed. It's actually called transcription. A copy of it is going to be made into this molecule called mRNA. mRNA is going to leave the nucleus and go to these things we said were ribosomes. So we'll get into a lot more of this later. You can look at these on the slides. I don't want to bog us down here because this is really what we get into with genetics, but this is a good preview of what that's all about. So what happens when ultimately, up here at this top image, you can see this is a growing amino acid chain. This would be a growing polypeptide that's made by this big double units. This is the ribosome here, and this is the mRNA. So stay tuned on that. Again, we'll get into this a little bit more later. And here's just a nice chart summarizing. And that's it.